Okay, so Nur Atika, can you mute yourself? I cannot start while Nur Atika is. Okay. Okay. Right. So, Assalamualaikum and good morning. Is my voice clear?
Yes.
Okay, thanks. All right. So today I'll continue with protein thermodynamics. In part 2, I will show you some example. Yeah. So yesterday I explained to you about the concept, about the basic concept of protein thermodynamics, and in this part 2, I will show you some example of protein thermodynamics, so it's easier for you to understand how to stabilize a protein. Yeah? Okay, so so when we talk about protein thermodynamics, basically we talk about protein folding. Yeah, but protein folding, how to stabilize the protein structure, so so that the protein can function. Yeah. So that's why when we talk about protein, we always put protein structure and function together. Yeah, because without structure, the protein cannot function. The protein has structure because it has to function. Yeah, without structure, the protein cannot function. So that's why understanding how to stabilize the protein structure is very important in studying the protein chemistry. Yeah. So that's why understanding how the protein folds and what factors that could impact the protein structure or the protein folding is very important. Yeah. So basically, this is the steps. Yeah, this is the steps, the mechanism how the protein folds. And at the end of this slide, I will show you some proposed mechanism. Yeah, there are different mechanism how a protein folds. Yeah. But basically, it starts from unfolded, which is inactive, yeah, and then it slowly folds, yeah, to get the structure until at the end of the process, the protein will adopt a folded structure, which is biologically active. All right, so I'm sure you still remember my introductory lecture about Unvinson's dogma. Yeah. So the dogma says that the three-dimensional structure of a protein can be determined from the sequence alone. Okay. Once we know the sequence of the protein, we will know the three-dimensional structure of protein, and this is the basis of protein structure prediction. So protein structure prediction means we can predict. Yeah, we can predict using computational method what the three-dimensional structure of a protein is without having to conduct an experiment. Because as you know, to get the three-dimensional structure, we can get the structure through X-ray or through NMR or through cryo-electron microscope. But in the absence of these three methods, we can still predict the structure of protein by knowing the sequence of the protein. Yeah? So the reason why Unvinson came up with this dogma because of this very simple experiment. Yeah, very simple experiment. So he started with a small protein. Yeah, the length of this protein is just over 100 amino acid. So this protein is considered as a small protein called a ribonuclease A, yeah, as a model for folding. Yeah. And he destroyed the structure using urea and mercaptoethanol. So what is the function of beta mercaptoethanol? Anyone? It's a yesterday's lecture. What is the function of beta mercaptoethanol? Okay, every time you're a chemist, right? Every time you heard something mercap, yeah, either mercaptan or mercaptoethanol, it has SH group, right? It has sulfur. Okay, so what is the function of beta mercaptoethanol?
Reducing agent.
Reducing agent of? Yeah, it's true. It's reducing agent.
Sulfate bond.
Yes, exactly. So the reason why he added beta mercaptoethanol is to break disulfide bond. Yeah. So as you can see here, there are 4 disulfide bonds. 1, 2, 3, 4. Yeah, disulfide bond. Okay. And these disulfide bonds stabilize the protein structure. And in order for him to destroy the structure completely, urea is not sufficient, because urea, the function of urea is to strip off the water from the surface of the protein. That's it. But even the water are stripped off from the surface of protein, there are still 4 disulfide bonds that keep the protein to its structure. So that's why he added beta mercaptoethanol to completely break the disulfide bond. So you see here, all the disulfide bonds are broken, so the protein can be defolded completely. Okay. And then what he did was he slowly removed the urea and beta mercaptoethanol by dialysis. Yeah. You know dialysis, right? Dialysis is you put the solution. Yeah. So you put water here, all right, and you put the protein inside the tube. Yeah, this is the tube, dialysis tube. Yeah, a dialysis tube consists of a membrane with certain pore size. So this is the protein with urea and beta mercaptoethanol, and over time, the urea and beta mercaptoethanol will diffuse out of the tube, will go to the water, yeah, and the protein stay inside because the pore size of dialysis tube is very small. You know the dialysis tube, right? Anyone never see a dialysis tube? Pernah buat kan dialysis in analytical chemistry? You all dah pernah buat dialysis kan? Pernah?
Tak pernah.
Tak pernah? Okay. Well, it's shocking. Yeah. Dialysis is a very common method. I don't know, because I never teach year 1, year 2 student. Normally I teach year 3 students, so I don't know what you learn in year 1 and year 2. So dialysis is a way to remove the small particle from a solution. Yeah. So dialysis tube has a certain pore size. Yeah, different pore size. So you can buy different dialysis tube with different pore size. So kalau you letak your sample dalam ni, for example, your protein, and it has some small molecule like urea and beta mercaptoethanol, you can remove them by soaking the dialysis tube with your sample in water, yeah, or in any buffer. Then the small molecule, the urea and beta mercaptoethanol will diffuse out of your solution. Yeah, it will go to water. Yeah, sebab apa? Sebab dekat water, dalam water ni dia tak ada urea and beta mercaptoethanol, so dia akan keluar. Yeah, dia akan keluar, and you have to keep changing the water until you can completely remove the urea and beta mercaptoethanol from your sample. Yeah? Because it will create an equilibrium. Yeah, it will create an equilibrium. For example, you start with 6 molar of urea. Yeah, when you first dialyze your sample, it will reach equilibrium around 2 molar. Yeah, 2 molar of urea here in the big beaker glass, and 2 molar of urea in your sample. Yeah, and then you have to replace the water. Yeah, you replace the water here with fresh. Yeah, with fresh water, and then you continue dialysis, and then it will reduce until 0.5. Yeah, and then you replace again with the new water, until you reach almost 0 molar of urea. Yeah? So that's dialysis. Basically macam you basuh baju. Kalau you basuh baju kan pertama you letak deterjen kan, ya, you letak deterjen, you kacau. Lepas tu you rinse kan, you nak buang the deterjen, apa you buat? You rendam dalam air kan. Lepas tu you kena tukar kan, sebab the first rendam, the first rinse, it will reach an equilibrium. See how chemists think, you know, even when I wash my clothes, I still think about chemistry. So when I rinse, yeah, with the first batch, yeah, so it will reach equilibrium. Yeah, it will reach equilibrium. So letak dalam baldi kan, letak dalam ni baju. This is the first rinse lah. Yeah, it will reach equilibrium. Yeah, so and then you need to replace the water, right, with the clean water, and then it will reach another equilibrium. You replace again and again until no more bubble you can observe in the water. But you have to replace it multiple times. You cannot just put once and then you can remove all the detergent, cannot, because somehow it will reach equilibrium between the detergent dekat baju ni dengan detergent dekat air. Yeah, it will reach equilibrium. If the equilibrium already reached, you cannot keep pushing detergent to diffuse out from your baju. Yeah, sebab dia dah equilibrium, so you kena tukarlah air tu. Sama dengan dialysis. Okay, dialysis dia punya principle macam tu. The first batch, once it reach the equilibrium, you kena tukar air dia. Second, tukar balik. So it's a long process basically. Yeah, dialysis is a long process, but now people replace it with HPLC. That is faster. Yeah. So with HPLC, we call it as size exclusion chromatography. Yeah, 3401 pernah dengar kan? Size exclusion chromatography. So we put this sample in a column, yeah, with certain resin, yeah, and then the protein will stuck inside, and the small molecule will go out of the column. So this process is faster, but during Unfinster's time, there is no size exclusion chromatography, so that's why he used a dialysis. We still use dialysis in the lab. I still use dialysis in the lab, you know, because you can leave, yeah, you can leave the solution, you know, for some time, you know, for a few hours, for several days, and then you can collect your sample free of urea and beta mercaptoethanol. All right. Okay. So go back to this experiment. So what he found out, yeah, after removing all urea and beta mercaptoethanol, and he did oxidation. He did oxidation to create the disulfide bond back. Yeah. Ini yang apa ujian 1 kan? I ask you about what is the mechanism to create the disulfide bond. Only 1 or 2 students can answer correctly. By air oxidation. This is the way to stabilize the protein. Yeah. So kita just purge with oxygen 3 jam, 6 jam, and then the disulfide bonds will form again. Yeah. So after doing this dialysis and air oxidation, he found out that he can get the structure back. So dia punya structure balik macam yang ni lah, macam yang initial structure. So it's amazing that how no matter how bad you are destroyed, you can always come back to your initial condition. Yeah, so that's amazing. So why this protein can go back to its 3-dimensional structure? Because of the sequence. Yeah, the sequence is still the same. So the protein will come back to its initial structure. Tapi tak selalunya macam ni, because in reality, yeah, well, he used very small protein, yeah, 100 amino acid sahaja. In my work, I use bigger protein, yeah, 500, 400 amino acid. Sometimes sometimes I cannot get the structure back, you know. So the Unfinster dogma actually is is beautiful in concept, but in reality, it's difficult to implement. Yeah, I have a very bad experience with big protein when I destroy the structure of the big protein, and then I remove the denaturant, yeah, the denaturing agent. I cannot get the structure back. Yeah, I don't know why. Until now, I don't know why. After several years, I still don't know why. But this is not just my experience. Many people they experience the same as well. Yeah. So this dogma, I would say, it's easier said than done. Cakap senang, tapi masa kita buat macam it's very difficult actually. Yeah. Yeah. So this is how he did the experiment lah. Yeah. But as I said, when you work with a bigger protein, then you might not get the initial structure back without the presence of chaperone. Yeah, if you work with big protein, you might need chaperone to help the protein back to its three-dimensional structure. Okay. Yeah. So this is what you answer correctly just now. The 2-beta mercaptoethanol is, you see this, it has SH, right? So it's used to disrupt the sulfide bond. So you can monitor, yeah, you can monitor the process of protein unfolding using NMR, yeah, to know that your protein is unfolded. Yeah? This is unfolded protein, right? You can see it using NMR, yeah. Other than NMR, you can also see it using CD, yeah, circular dichroism, which I will explain to you next week. Yeah, so next week I will explain to you different methods to see the protein structure. So NMR is more complicated, yeah. The easiest one is by using CD, ya. Sebab CD ini dia macam UV vis. You letak aja dalam machine, you can see the reading, you can see whether your protein is folded or unfolded. We have it in UPM, yeah. Only one instrument. We bought it when I was here in 2009, yeah. So we put it in Faculty of Biotech. So they routinely use circular dichroism to see the integrity of protein structure, to see whether the protein is destroyed or whether the structure is still intact. NMR is more complicated because the spectrum is more complicated to interpret, but CD is easier to interpret. It's just like UV vis, ya. Senang nak faham dia punya spectrum. Okay, other than in vitro, yeah, in vitro you use the machine, yeah, to see whether the protein folds or not, yeah. Other than that, you can also do the in vivo experiment, yeah, to study the intracellular protein that assists folding. Yeah. So in this case, of course, you need chaperonin, yeah, to help the protein to fold. So I'll skip this. This is how we study the process. Yeah, we can use a different spectrophotometer. I personally prefer circular dichroism, but as I mentioned, the information that we get from circular dichroism is too simple. Other people, they use differential scanning calorimetry, DSC, to study the steps when the protein folds or when the protein unfolds. So this one I will discuss in next week, yeah, when we discuss spectrophotometer techniques to see the protein structure. Okay, this is something that I asked you yesterday. If you boil egg, can you unboil? Yeah, telur dah keras kan? Can you unboil? Some people say no, we cannot unboil. Yeah. Buat apa nak unboil kan, terus makan je kan. But this scientist, yeah, from University of California at Irvine, he said it's possible. Yeah, it's possible by doing something. Yeah, you can refer to his paper to see how he unboiled the protein. Yeah. So basically, it's possible, but of course, it's not as simple as boiling the protein. Yeah, because it's already hard, but it can be done. Yeah, theoretically it can be done, and practically it can be done. Basically all proteins, yeah, as long as you don't destroy you don't cut the chemical bond, yeah, all proteins actually, once you destroy it, you can bring the protein back to its initial structure. Theoretically can, yeah, you just have to find the way how to do it, because chemically it's still intact. Ya, tak ada chemical bond yang you potong. Ya. So basically you still have the intact protein. It's just how you arrange it. Now it's different. Yeah, the arrangement, the three-dimensional arrangement is different. So basically, theoretically, yes. Yeah. If you can find a way, yeah, to bring the protein back to its initial structure, then basically we can solve the Alzheimer's disease. We can find a treatment for Alzheimer's disease, for Parkinson's disease, for CJD disease. Because all these diseases are caused by unfolded protein. Yeah, you can rearrange the protein to go back to its three-dimensional structure if it's outside your brain. Masalahnya ada dalam otak. And not many things can enter your brain. Brain is the most inaccessible organ in our body. Ya, memang susah nak access brain, ya. Because God created brain as something that is protected by your skull, yeah, by your skull, and also it's protected by something that we call blood-brain barrier. Yeah, 3B, yeah, blood-brain barrier. Even blood cannot enter our brain. That's how protected our brain is. It's completely protected. Yeah, the most inaccessible organ in our body. Even virus and most virus and bacteria cannot enter our brain. Okay, because of blood-brain barrier. Yeah. So that's why it's very challenging, very challenging when you have something in your brain, either it's tumor or his disease. It's very challenging to treat it. Bukan senang. Ya, kalau you ada tumor, ada disease in other parts of your body, it's easier to treat sebab dia orang accessible. If you have a tumor in your lung, you have tumor in other parts of your body, so you can easily get the drug to go to that area. Tapi otak ni susah, yeah? Because it's well protected. Yeah? So that's why in the past, we can detect someone with Parkinson's when that person died. So we open the brain and then we stain it. Yeah, we stain it using spatial stain, and we know, okay, it's because of Parkinson's disease, because of some unfolded protein. But now, scientists come up with some molecules that can penetrate blood-brain barrier and enter the brain and can detect the presence of unfolded protein. Yeah, but it's very challenging. It's very challenging. Some virus can enter the brain. Yeah, macam Japanese encephalitis virus, meningitis, it can enter the brain, but not all. Not all virus. Yeah, so thank God that our brain is well protected. But one day, I mean, if one day you are able to figure out how to bring drug enter the brain, yeah, and untangle the unfolded protein and bring the protein back to its initial structure, you can cure Alzheimer's, you can cure Parkinson's disease, basically. So this is something that people look forward to. Yeah. So think about it. Yeah. When you graduate from UPM, you are interested in solving this long-standing problem. Yeah. So that would be good for everyone. Again, brain is the most difficult organ to work with. Very difficult because of the level of protection that we have in our brain. All right, so that's Unfinson dogma. What about Levinthal's paradox? Yeah, you still remember, right? Levinthal's paradox. So Levinthal's paradox says, if we let the protein to fold by itself, it will take time longer than the age of universe. Kita ni semua tak ada. If the protein folds by itself, without any help from anyone. Yeah, that's why it's impossible. That's why protein, in order for the protein to fold, it needs help. It needs to undergo certain mechanism. Yeah, it's impossible it folds by itself by random mechanism. Impossible. Yeah. So protein folding is not random. Yeah, it must have pathways or helper, something to help the protein to fold. Yeah. So this is something that Levinthal challenged the Unfinson dogma. Yeah, Unfinson says, well, if you have, as long as you have the sequence, the protein will fold to get the three-dimensional structure. But Unfinson did not say how. He just say, as long as you have three-dimensional structure, as long as you have the sequence, it will form the three-dimensional structure. And Levinthal came by saying that there are too many possible conformations. Too many. It's impossible that the protein can have certain structure without certain mechanism or without any help. Yeah, so that's why Unfinson and Levinthal, they complement each other. Yeah, what Unfinson says is correct. Yeah, but Levinthal came with question, how? You senang je cakap, but how? Okay, cakap senang, but how? Okay, so that's why Levinthal asked this question, and we call it as the Levinthal paradox. So the question posed by Levinthal, yeah, the question asked by Levinthal, it motivates scientists to look for specific folding intermediate. So scientists came up with different mechanisms. Yeah? With different mechanism, how the protein folds. Yeah? So at the moment, there are three classic models of protein folding. Yeah. So different scientists propose different model. Yeah? So basically, the protein starts as a framework. Yeah. So it has some structure in the form of framework. Yeah. So dia bukan complete structure. Yeah. But it has some loops. It has some initial folding. Yeah. So the protein starts with this structure. And then once the protein starts with this initial semi-folded structure, it can undergo three mechanisms, either diffusion collision, propagation, or hydrophobic collapse. Yeah? So we will see one by one. All right. Okay. So what framework model is? Yeah. So based on Kim and Baldwin, they said once the protein is released from ribosome, yeah. So this is the ribosome. This is the ribosome. This is the protein. Yeah. Once the protein is released from ribosome, the protein will try to fold by itself. They try to fold by itself, okay, to form the secondary structure, macam apa alpha, beta, yeah. So it will start to form, yeah, macam baby kan, macam baby dia start to jalan, tapi dia tak perfect, atau dia still apa, is still cannot walk perfectly. Yeah. So it starts with the formation of secondary structure elements. Yeah. And then all these elements, they interact to form a more advanced folding intermediate. So it starts with a primitive structure. Yeah. A primitive structure. And then once this primitive structure is formed, it will undergo three mechanisms. Yeah, either through diffusion, propagation, or hydrophobic collapse. So we'll see one by one. This is the first mechanism, diffusion. Yeah. So this mechanism says, or it proposes that if you have this primitive structure, initial structure, they will diffuse, they will collide, yeah, to give a bigger structure. So this concept is proposed by Martin Karplus and Weaver, yeah. So they will just collide, just like our universe, yeah. In the beginning of our universe, semua panas kan, semua panas, and then they start diffuse and collide. And then we see planets, we see stars because they collide and diffuse. Yeah, so Karplus and Weaver use the same concept to explain why we have protein with certain structure, because so we start from a disordered structure, and then they collide and diffuse. Yeah, they langgar each other and then they fuse, and then they form a perfect structure, just like our universe. Yeah. Second concept is classic nucleation. Yeah, classic nucleation ni macam in organic chemistry, you pernah buat ni tak, crystallization. You crystallize the salt. Yeah, so you have a solution. You put this is a saturated solution of salt. Yeah, it's saturated. You nampak masa crystal tu nak start, you can see this very small initial crystal, yeah, and this small crystal will grow bigger and bigger to form bigger crystal. Okay, this is my most favorite experiment because I like to observe. I observe the formation of small crystal, and then I leave it for several hours, several days. It gets bigger and bigger. All right. So this is the classic nucleation model. So basically, this model says the framework structure, initial structure, disordered structure, it has different structures. Yeah, it has different structures. Okay, dia tak satu structure je, dia ada beberapa structures lah. And then the structure that is closest to the final structure will become the nucleus. So dia ada beberapa structure lah, ni 1, ni 2, ni 3, 4, 5, 6, 7, etc. The structure that is closest to the final structure will become the nucleus. For example, this structure is the closest with the final structure. And then other structures, they will change the structure. Dia akan tukar structure dia to become this structure. And then they grow bigger and bigger. Macam the crystal grow lah. Kalau you nampak crystal kan, dia grow. It starts from one small crystal and then it will grow and grow. All right. So that's a classic nucleation model. The third one, this is my favorite model. Yeah. So when you have this framework structure, they will collapse. Yeah. The way they collapse is they will put all the hydrophobic amino acid inside the structure. Yeah, semua amino acid yang hydrophobic dia akan letak dekat dalam, and then the hydrophilic amino acid dia letak dekat luar. Because what? Because dekat luar ni semua air kan, H2O. So the protein will arrange themselves to put all hydrophobic amino acid inside in the internal part, and the hydrophilic amino acid outside on the surface. Okay, so this is the most realistic model based on my opinion lah. So that's why I told you I like this model, because this is the most realistic and the easier to understand. Okay, but in reality, yeah, it is unlikely that there is a single mechanism for protein folding. Sometimes they combine. They combine nucleation and condensation. They combine nucleation and hydrophobic collapse, or they combine all three together. For example, this protein, yeah, Barnes and Barstar, these are two proteins that they join together. So Barnes and Barstar ini dua protein, ya. Two different proteins, they join together to function. Yeah? Basically, the mechanism is they combine three mechanisms. Yeah, nucleation, nucleation, ada, hydrophobic collapse pun ada. Yeah, and then another mechanism apa? Collision. Yeah? Collision and collision and diffusion. Yeah? So they combine three mechanisms together to form a Barnes and Barstar complex. So this is how the protein looks like. Yeah? So this is the Barnes, yang besar ni. Yang kecil ni is Barstar. Yeah? Two small proteins to fold and fit together. This is another example. Lysozyme. Lysozyme is small protein. Yeah? They fold by a different pathway. Yeah, by different pathway. So the way they fold is they form alpha helix first, followed by beta domain. Then they fold. Yeah. So the way they fold basically based on the hydrophobic collapse model. There are different factors that affect protein stability. As I mentioned yesterday, pH, yeah, is very important. That when you put the protein in different pH, structure dia akan bertukar. Why? You have to think about it. Yeah? When I ask in the exam, when you change the pH, why the structure change, then you need to come up with some mechanism. Senang je nak faham. As long as you know what pH is, you know the structure of protein, yang hydrophobic amino acid ada dekat mana, hydrophilic amino acid ada dekat mana, and you can think of the effect of pH on the protein stability. Yeah. So you have to be able to think beyond the lecture notes. Yeah, beyond the lecture notes. So that's why having a textbook is very important. Yeah, I keep telling my students have a textbook because the detailed explanation in the textbook. PowerPoint is just points. Yeah, you read the PowerPoint, you don't get the whole concept. But if you read the textbook, you get the whole concept. If I ask you the question, how pH affect the protein structure, you cannot find that in my slides. No, cannot. But if you read the textbook, they will explain to you how the pH affect the protein structure. Okay, so you can expect this kind of question during your test 2 and final exam. More analytical question requires you to think deeply, you know. So yeah. Ligand binding also affects the protein stability. Yeah, if you have inhibitors, yeah, it will affect the protein structure. If you have iron, it will affect the protein stability as well. There are many proteins that we know it can only function if there is a metal ion. For example, hemoglobin. Yeah, hemoglobin is protein yang ada dalam darah kita. Yang kerja dia is to transport the oxygen. Yeah, dia kena ada ferrum, ya, kena ada iron in order for the hemoglobin to work. And then disulfide bond, yeah. This is I already show you before that many protein they rely on disulfide bond to maintain the stability. When you add reducing agent like Mercaptoethanol, it will destroy the disulfide bond and it destroy the protein structure as well. Solvent, yeah, solvent is very important to stabilize the protein, yeah. I show you yesterday how the protein how the water stabilize the protein. Ini water, yang kecil-kecil ini water, and this is the protein. As you can see, yang kuning ni hydrophobic group. Hydrophobic group, they try to avoid water, right? So that's why dia berkumpul dekat dalam ni. Yeah, they gather inside in the interior part of the protein sebab dia orang tak suka dengan air. Tapi yang dekat luar protein ni is hydrophilic amino acid, yeah, like arginine, aspartic acid, glutamin, glutamic acid, dia orang ada dekat luar. Sebab apa? Dia orang ni polar, polar and hydrophilic amino acid, so they like to interact with water. So that's how protein organize themselves. Semua yang hydrophobic, they will collapse, they will gather in the internal part of the protein. If you change the solvent, it will change this structure. Ya, contoh, you tukar air ini kepada hexane. Apa yang akan berlaku? Kalau if I ask you during the exam, if you change water with hexane, what will happen? What will happen with the protein if you change water with hexane? Anyone?
The structure change?
Yes, how?
Not quite sure.
Yes, the hydrophobic group will be on the surface. Yeah, thank you. So this hydrophobic group will, because hydrophobic group, they like hexane, right? And hydrophilic group, they don't like hexane. The structure will flip. The structure will flip. Yeah, so basically you destroy the protein because the structure flips. So initially the hydrophilic group on the surface, hydrophobic group is in the internal part of the protein, and because you change the solvent with hexane, now protein itu terbuka. Yang dekat dalam dia keluar, yang dekat luar dekat dalam. So basically you destroy the protein completely. Yeah, so that's correct. Yeah, so when you change the solvent, yeah, it will affect the protein structure. If you change the pH, it will affect as well. Yeah. If I ask you what is how pH affect the protein structure, okay, so you need to really understand about the concept of pKa. Yeah, because if you have this protein, okay, you have, for example, you have aspartic acid. Yeah, aspartic acid is COOH. Yeah? This is one example. In acidic pH. Yeah, in acidic pH, it will stay as COOH. COOH is natural, right? Betul kan? Tak ada charge, But then, if you increase the pH, apa yang akan berlaku? You increase from 5 to 9. What will happen? You have more OH minus here. What will happen? Correct. Yeah, deprotonation of COOH. So this OH will disappear. Yeah, it will become COO minus. So now you have charge in your protein at high pH. Yeah, at high pH you have charge in your protein, and it will impact the whole structure of the protein. Yeah, so that's how pH affect the protein structure. So this kind of question that you can expect in your second and final exam. So you need to understand how pH affect the protein structure. It means you need to understand about how pH deprotonate the carboxylic acid group. So yeah, basically I expect all the chemistry graduates to have this kind of thinking skill. Not textbook thinking skill, but analytical thinking skill. Yeah, by combining all chemistry branches, organic chemistry, analytical chemistry, physical chemistry, because at the end of the day, when you work in a company, you don't know which chemistry you need to apply to solve the problem. Oh, this is the problem. Which chemistry I need to apply to solve this problem? Is it analytical chemistry? Is it organic chemistry? Or is it physical chemistry? No. It's chemistry, So to answer this question, you need to combine all chemistry branches. Yeah. So protein chemistry actually requires you to understand all the chemistry branches that you learned before. Yeah. Some organic chemistry is required to understand this issue, some analytical chemistry, yeah, especially the concept of pKa. You know, you need to understand the concept of pKa, the concept of pH, in order for you to answer this problem. Yeah. So yeah, this is the reality that you need to face when you go to your third year, fourth year, and eventually when you work. You don't know which chemistry you need to solve the problem. What you know is you need chemistry. That's it. You need to combine all chemistry branches that you learn during the past 2 or 3 years. Okay. All right. So that's all for today. Yeah. I hope you have some basic understanding about protein stability. You know, about protein thermodynamics. For you to you know to in the future, yeah, when you work with a protein, yeah, in a pharmaceutical company, then this concept is very important. When you face a problem with instability of drug, instability of vaccine or antibody, then you might need this concept to help you to solve the problem. That's all for today. Any question before we dismiss?
No.
Okay, if no question, then that's all for today. I upload the lecture notes